physical unit of heredity, a linear sequence of nucleotides along a segment of DNA that provides the coded instructions for synthesis of RNA, which when translated into protein leads to the expression of hereditary character.
Gene expression, the conversion of the information encoded in a gene first into a messenger RNA and then to protein.
DNA, deoxyribonucleic acid, an extremely long macromolecule that is the main component of chromosomes and is the material that transfers genetic characteristics in all life forms.
Uracil, a pyrimidine base that is one of the fundamental components of RNA in which it forms base pairs with iodine.
mRNA, a single-stranded molecule of RNA that is synthesized in the nucleus from a DNA template and then enters the cytoplasm, where its genetic code specifies the amino acid sequence for protein.
tRNA is a small RNA molecule consisting of strands of nucleotides folded into cloverleaf shape that pick up any unattached amino acid in the cell cytoplasm and conveys it into the ribosome for protein synthesis.
rRNA, a type of RNA distinguished by its length and abundance, functioning in protein synthesis as a component of ribosomes. RNA polymerase, an enzyme that synthesizes the formation of RNA from a DNA template during transcription.
Transcription, the act or process of transcribing. Translation, the process of converting the information contained in a sequence or RNA bases into a sequence of amino acids.
Codon, a triplet of adjacent nucleotides in the messenger RNA chain that codes for a specific amino acid in the synthesis of a protein molecule.
Anticodon, a sequence of 3 nucleotides in a region of transfer RNA that recognizes a complementary coding triplet of nucleotides in messenger RNA during translation by the ribosomes in protein biosynthesis.
Ribosome, a tiny, somewhat mitten-shaped organelle occurring in great numbers in the cell cytoplasm, either freely in small clusters or attached to the outer surfaces of endoplasmic reticula, and functioning as the site of protein manufacture.
Elongation, the act of being elongated or the state of being elongated.
Peptide bond. Now, a covalent bond formed by joining the carboxyl group of one amino acid to the amino group of another with the removal of a molecule of water. Stop codon. Now, a codon that stops the synthesis of a protein molecule.
Promoter region, a region of the DNA that facilitates the transcription of a particular gene. Cystic fibrosis is a hereditary disorder characterized by lung congestion and inflammation by malabsorption of nutrients and the pancreas.
Transcription of CF, transmembrane conductance regulator, requires a CCAA-T-like element for both basal and CAMP mediated regulation.
In a nonsense mutation, the new base changes a codon that specified an amino acid into one of the stop codons. This will cause translation of the mRNA to stop prematurely and a truncated protein to be produced.
This is a chart showing the inheritance of cystic fibrosis. A child has a 1 in 4 chance of actually getting the cystic fibrosis disease when both father and mother are carriers of the gene. They have a 2 in 4 chance of just becoming a carrier of the gene. This picture is 2 long airway passages. To the left is someone without CF, and to the right is someone with CF. The left picture without CF shows a lot more circulation throughout the lung, and the one to the right shows a lot more bacterial mucus because there's not as much hydration. So our product helps reactivate the CFTR channel with patients with CF, which helps more water come in the lungs and helps clear out most of the mucus.
Like we stated previously, our pill helps get more water flow in the lungs and decrease the bacterial mucus, making it easier to breathe and making your lungs healthier.
Yeah. Mutated and normal CF sequences.
Reverse engineered DNA sequence.